what if I told you that we are all sitting here today thanks to these microscopic organisms that you can't even see with your bare eyes that live in the ocean? These are phytoplankton. You can see them here in a picture under the microscope. They live in the scale of microns, and you can think about them as tiny plants in the ocean and in other aquatic ecosystems. They're actually responsible for half of the photosynthesis that takes place on Earth. So you can thank these guys for every second breath you are taking. And despite being so small, they can generate large-scale blooms that can even be seen from space, like in this picture taken by a NASA satellite near the coast of Yemen, which is now notoriously known for other reasons. And these beautiful colors are not generated by an AI or something like that. This is basically what happens when these cells reach high concentrations and change the color of the sea, and this can be seen from space. And just like plants on land are the basic of food webs, also phytoplankton or algae in the ocean are the base of marine food webs. They are supporting the growth of all marine life. And these giant blooms are also providing major food source that is entering the system. So not only they are important for the marine life, but also for biogeochemical cycles of different elements, such as the carbon cycle, and they can have also important effects on, important impacts on climate. And these blooms are characterized by a rapid proliferation of the population, reaching high densities, as you can see with these greenish colors. But they also collapse in what seems to be simultaneously across very large scales, considering that they are only microns in size. And it's still unclear what coordinates this demise and what leads to the mortality of these organisms. And this is something that we're interested in studying in the lab. And why should we even care about some algae dying somewhere? So first of all, they have an important ecological role, and we want to understand better the bloom dynamics and their impact on the marine ecosystem. But also some of these blooms are toxic, and they can be toxic to humans and to other animals, and they have major hazardous effects on health and on economics. So there is a lot of interest in understanding when is the toxin going to be released, when is the bloom going to be terminated, whether we can predict these bloom events. And also, some of these algae are grown for biotechnology applications in agricultural facilities like this one in Ktora. And we want to be able to monitor our cultures and to make sure they are healthy, to prevent their collapsing. And that leads us to different applications of understanding the mortality of algae. And in the lab, we want to try and zoom in from this ecological large phenomenon to understand it with molecular cell biology approaches. And one of the mechanisms that was suggested to be involved in the demise, as well as in the general rapid turnover of these organisms, is through programmed cell death, which is a regulated and active form of cell death. And it was suggested that different environmental stress conditions, such as viral infection, bacterial pathogens, nutrient limitations, could lead to the induction of cell death in these cells. But we still don't know what is the molecular machinery behind it, and we don't know what are the genes that participate in it, and they are lacking most canonical machinery known from programmed cell death in animals. For example, the apoptotic machinery is missing. And in animals, we know that programmed cell death is very important for maintaining the general homeostasis of the organism, for prevention of cancer, for the development. But in unicellular organisms, it's much harder to understand how such a little trait evolved. And if you think about it, how could you evolve a trait that actually kills you? One potential explanation could be that the death of part of the population enables the improved survival of the rest of them. And I was interested in understanding how can some cells survive stressful conditions that actually kill most of the population. And if you think about it in the context of the bloom, during the demise, almost the entire population dies, but some cells are able to survive and serve as a seed for the next bloom. And in order to study these processes further, The aim of this project was to identify the genes that are involved in this algal cell death. And previously in the lab, we discovered that in response to stress, cells from the same culture that are genetically identical respond differently. And when I'm talking about stress, I'm not talking about the stress of balancing life and work. It's not that. They are stressed by different environmental stresses, as I mentioned, and in this system, we were looking into oxidative stress, such as highlight or hydrogen peroxide, and we noticed that some cells are highly sensitive to the stress and eventually die, while other cells are more resilient and are able to survive this stress. And we have developed a method in which we can predict what are the sensitive cells and what are the resilient cells when they are encountering this stressful event, this hydrogen peroxide treatment. And we did that using a fluorescent probe that basically reports the oxidation state within the chloroplast. This is where photosynthesis takes place. And it basically reports us the physiological aspect of the cell. And using this sensor, we can predict, based on the early responses of the cell, who is going to survive and who is going to die. And I aimed to use this system in order to identify genes that are involved in cell death or stress resilience. So I treated the cells with hydrogen peroxide in a dose that kills some of the population. And then using this fluorescent sensor, this protein that shines differently in sensitive and resilient cells, in order to sort these populations and explore the gene expression patterns in each of them using transcriptome analysis, basically to see which genes are induced in cells that are going to die and which genes are induced in cells that are going to survive. And the nice thing about this system is that both of these resilient and sensitive cells are coming exactly from the same culture. They are genetically identical, and they're experiencing the same stressful conditions. They come from the same treatment. So we can discriminate general stress responses from those linked to their specific contrasting phenotypes of either cell death or stress resilience. And indeed, when looking into the gene expression patterns, we saw that sensitive and resilient cells are at complete distinct states. This heat map represents the differentially expressed genes we discovered. Each row is a single gene, and each column is a single sample, and they're arranged based on their similarity in patterns. And we detected a cluster of genes that were specifically induced in resilient cells, and we suspect them to be involved in survival, while another cluster of genes was specifically induced only in cells that were going to die, and these form candidates for cell death. However, we have hundreds of genes, and we needed to select a few in order to study them more in depth, and we still don't have a way to study in my algae of interest all of these genes simultaneously. So we looked into another algae, another system in which recently a very large-scale mutant screen was performed. They screened tens of thousands of mutants over more than 100 conditions, and this data set gives us an input about the function of the gene. So I was looking for genes that in my subpopulations were induced specifically in cells that were going to die, and that the same gene, when mutated in the other algae, gave a resilient phenotype, meaning that if you mutate this gene and you damage this PCD machinery, this cell death machinery, you might get less cell death, and you might get a more resilient phenotype, which is a very rare phenotype. And out of those candidate genes, we found cathepsin X, which is a cysteine protease. It's a protein that cleaves other proteins. And together with my Sader from the lab, we further investigated this gene and looked into mutants in both of these algae. And these algae are very distant from each other. Basically, this one is even closer to plants than to the algae I was working with. And we discovered that mutants in cathepsin X in both of these algae are more resilient to oxidative stress in the form of hydrogen peroxide. You can see on the y-axis the cell death, the mortality, and on the x-axis the different doses of hydrogen peroxide. And both of these mutants had lower mortality compared to the wild type. And this led us to the conclusion that cathepsin X is a conserved function in cell death. And it's actually so conserved that even we have this protein. Cathepsin X is conserved from human to algae and probably evolved early in the evolution of eukaryotes. And with the help of Orly Dim, we generated these structural models of the human, protein in gray, and in purple the phytoplankton, the algal protein. And you can see that they are very similar, and if you zoom in into the active side, the part of the protein that actually has the activity, they are very similar to each other. And in humans, this protein was shown to be involved in various diseases, from cancer to neurodegenerative disorders, and we still don't know whether it plays a role also in regulated forms of cell death. So to summarize, I hope I convinced you that phytoplankton are important, and to show you that we were able to identify genes involved in sulfate regulation using the variability in the population in response to environmental stress. One of them was cathepsin X, which is a conserved cell death protein that we novelly discovered. And we hope to use the genes that we discover in order to develop sensitive biomarkers for different cellular states, so we could detect cell death in the environment or possibly predict it in environmental conditions, maybe even in toxic bloom. And with that, I would like to thank you for listening and thank the organizers and Asaf Vardi, the amazing Vardi Lab, and especially my Sadeh and all the people who are involved in this project. Thank you.